Hi, my name is Lydia Jones, and my presentation is on the genetic history of an archaic hominin group from the Denisova cave in Siberia. So this study falls under the field of paleogenomics. And what is paleogenomics? Well, it's the extracting and characterizing of genetic material from ancient and/or fossilized remains. So DNA from ancient specimens is referred to as aDNA or ancient DNA, and due to the natural degradation that occurs in DNA over time, aDNA has to be treated a little differently than modern DNA. So how did the field of paleogenomics get started? Well, the first extraction of ancient genetic code was in 1984, and it was DNA sequences from a species called a quagga, which is an extinct equine species. And scientists were able to perform this sequencing because they had access to a dried muscle sample of a quagga, and they were able to recover 229 nucleotide pairs. And when they compared that to modern equine species, they showed that it was very closely related to modern-day zebras. And what this study did was it showed that it was possible to get genetic material from extinct animals. And this really opened the door, the treasure trove to the field of paleogenomics. And other significant studies since then included the first entire genome sequencing from Homo neanderthalensis, which occurred in 2013. The material was taken from a finger bone, and it was conclusive evidence for Neanderthals and humans interbreeding. So that was something that had been a really big debate among the scientific community for quite some time, and this confirmed that they had in fact interbred. And it's estimated that up to 20% of the Neanderthal genome has survived in some modern-day humans. Another really interesting case was the Kanoic man, who's a 9,000-year-old Paleo-American man that was found in Washington State. And there was a lot of debate for some time over what his origins were, if he was European in ancestry or Native American. And there was kind of a custody battle for the remains. And it was discovered via genomic testing that he was, in fact, more closely related to modern Native American groups than to any other ethnic group. And so custody was given to 5 tribes that were local to the area. And as of 2016, the remains were given a tribal burial. And so the specific study that I'm going to be covering is the discovery in the Denisova cave, a study from 2010. The Denisova cave is in the Altai Mountains of Siberia in what is modern-day Russia, and it was a cave that had actually been excavated for quite some time, 20 or 25 years, and they knew the cave pretty well. They knew that it was a site of Paleolithic human occupation, so we knew that Homo sapiens had been living there. But when they went a little deeper down into the soil, they found a 40,000-year-old finger bone from a juvenile female, and then they also found the tooth of a young adult a little bit deeper in the soil. And they identified these specimens as being from a completely new hominin species, which they dubbed Homo denisova. And they did take genetic material from these remains, and the first step in doing that was basically to cut off the inner part of the finger that they found, of the finger bone that they found. So the thing about ancient DNA extracting DNA from ancient bones or fossils, is that it is a destructive process. So that's something that has to be taken into consideration, and it's not something that's taken lightly, because once you do that, it's kind of a one-shot. You know, you've used up that material, and you're not getting it back. So it's definitely something you want to be really careful with, and do the best that you can on the first try, because that's all you get. So all procedures were performed in a clean room to minimize possible cross-contamination, to really minimize the risk of that, so they would get good results the first time. So what they did was they treated the DNA with 2 enzymes, uracil DNA glycosylase and endonuclease 8. And what the uracil DNA glycosylase did was it removed uracil residues from the DNA and to create a basic sites, which are basically sites that neither have a pyrimidine or a purine. And then what the endonuclease 8 did was it cut at those from 5' to 3' at those abasic sites. And then they followed that with incubation with T4 polynucleotide kinase and T4 DNA polymerase to generate 5' phosphorylated blunt ends so that they could perform adaptor ligation. And adaptor ligation is just essentially the addition of synthetic nucleotides to a DNA strand. So after that, they created 2 separate sequencing libraries. And a sequencing library is just a collection of DNA fragments that have been stored and propagated via molecular cloning. So that DNA that they took earlier, that they processed with enzymes, and then those fragments that they obtained, they then inserted into cloning vectors, and then those bacterial cells propagated, which propagated the DNA. And then with those sequencing libraries, they used PCR, the polymerase chain reaction, to add a 7-nucleotide index to the prepared library molecules. And the purpose of that was just to prevent cross-contamination from other libraries in the lab, so to reduce the chance of mixing occurring. And those libraries that they had created were then sequenced using a platform called the Illumina Genome Analyzer. So what they were able to do with this genetic sequence was to compare it to other extinct hominids, specifically Neanderthals, and then current modern-day humans. So they took the genome from the Denisovan specimen that they had found, and then they compared it to 4 Neanderthal individuals, one from Mezmaiska Cave in Russia, and then 3 from Vendaja Cave in Croatia. And then they compared that to 5 human population groups, so the San, the Han, the Papuan, the French, and the Yoruba peoples. And so between this, they were able to estimate the divergence between these pairs of ancient humans and these modern humans. And they found that the DNA sequences of the Neanderthals and the Denisovan individual diverged approximately 640,000 years ago, and then from present-day humans around 804,000 years ago. So this means that Denisovans and Neanderthals split off around the same time from the human lineage, and then separated from each other, so they would have been more closely related to each other than they would have been to humans. So another thing I mentioned was that they found a tooth as well, in addition to the finger in the cave, and the tooth was from a different individual than the finger was, and they also did genetic analysis on that. So they used a dentistry drill to extract material from the root of the tooth, and then they isolated mitochondrial DNA from that. And they were able to identify a total of 15,094 sequences, and they compared that to mitochondrial DNA that was sequenced from the finger by a different group of researchers, and they discovered that they both differed at about 380 positions from both Neanderthals and present-day humans. And they concluded the tooth and the finger were from 2 different individuals, like I said, but in the same population. So they were both Denisovans. So what this tells us is that the Denisovan population is a sister group to Neanderthals, but they did have some archaic DNA that neither humans nor Neanderthals had. And this could be due to the fact that Denisovans simply retained this archaic mitochondrial DNA, and humans and Neanderthals did not and lost it in the course of evolution, or they could have possibly interbred with another unknown, more archaic hominin that we just haven't discovered yet. And so Denisovans don't appear to have contributed genes to modern-day humans in Europe and Asia as much as Neanderthals did. So we obviously know there's quite a few humans that carry a significant amount of Neanderthal DNA, but does not appear to be the same case with the Denisovans. So far, the Denisovan group has been identified as a distinct population. It's obviously very different, both morphologically in the characteristics of the bones and genetically from either Neanderthals or humans, but so far it does not have a formal Linnaean taxonomic designation that would officially separate it from Neanderthals or humans, genus or species-wise. but like I mentioned earlier, the unofficial name that some scientists call it is Homo denisova. So there have been further studies done with Denisovan DNA, and they found that Melanesian populations do have a significant amount of Denisovan DNA, as well as some populations in Papua New Guinea. And then they also found, very interestingly, that some Tibetans have a hypoxia pathway gene, so a gene that has enabled them to be able to acclimate to higher elevation climates with less oxygen, enabled them to breathe better, that is believed to have been inherited from Denisovan ancestors. So obviously this implies that there was intermixing between Denisovans and humans. Or even that the intermixing between the Neanderthals and Denisovans, then the Neanderthals could have carried some of that Denisovan genetic material, and then when they interbred with humans, some of that was transferred that way. So there's been other discoveries that of interest in the Denisova cave. So far, no complete Denisovan skeleton has been found. We only have bits and pieces. A total of 4 individuals have been identified in the cave, and they've been identified from molars and a partial mandible, and then actually a 5th specimen was found. That was a leg fragment, and it was identified to be from a Neanderthal-Denisovan hybrid. So it's proof that Neanderthals and Denisovans did interbreed at some point. And we've also found artifacts, including rings, beads, pendants, tools, and weapons, some of which are pictured here. But it's unknown if they are human, Neanderthal, or Denisovan, because we know that all 3 at some point have lived in this cave throughout the course of history and prehistory. So they were not entirely sure which one these tools were made by. And that concludes my presentation. Thanks for listening.